Hi, everybody. My name is Lindy Dodd, and I'm a research biologist for the U.S. Army Corps of Engineers. And I'm currently participating in a long-term training development program where I'm working on my PhD at the University of Rhode Island, and I'm working in Dr. Meyerson's lab. And specifically, the work that I'm doing is related to invasion ecology. The study system is a aquatic plant called water chestnut. And so the purposes of today's presentation is to give you a little more information about water chestnut, some of the botanical terminology, some of the morphology terminology, a little background about the plant itself. And many of you are going to be participating in a harvest in the upcoming weeks for an experiment that I have going on, looking at hybridization, potential hybridization between two different species of water chestnut that have been introduced to North America, specifically the northeastern United States. And so there's some concern about what might happen if these two species do hybridize. And so I'll be elaborating a little bit on that, but mostly just giving you a background on water chestnut. So water chestnut is the common name for this plant. The genus is Trappa, and the two species specifically I'm going to be talking about are Trappa natans and Trappa bispinosa, and the VAR is variety Inumai. So water chestnut is found in the Lathuriaceae family, and it's native to Eurasia and South Africa. If you look down on the world distribution map of this is specifically for Trappa natans. You've got this green circle here, and that's where water chestnut or Trappa is native to. So that's where you would find it naturally, but it has escaped through different, I'm assuming, introduced into North America. Specifically the northeastern United States. There are actually reports of individuals bringing the plant from Europe and other places to the Northeast, and then introducing the plants, and then the plants escaping, and then they make it into our waterways, our water bodies. Lake Champlain is in Vermont. It's been infested with Trappa natans for a long time. The Hudson River, Connecticut River, and even in Rhode Island, you find Trappa natans, and it is spreading. So it's native to Eurasia, and it's considered invasive in the US and up into Canada, but I'm specifically going to be talking about any plant populations that were introduced into northeastern America. All right. The northeastern US. It is actually considered rare or endangered in Russia and Europe, and that's mainly due to development and degradation of like natural waterways, rivers, wetlands, systems. And it's cultivated as a food in China, and I'll show you why here in a little bit. The taxonomy of Trappa is very messy. Some taxonomists or botanists think there's only one species of Trappa, and that there are two varieties: Trappa natans, variety natans, and Trappa natans, variety bispinosa. But there are others that differentiate the speciation based upon slight differences in morphology, and they think there are 20 plus species. But research in the last 10 to 15 years as a result of advanced genetic analyses has indicated there are more than one species, and that they do have the potential to hybridize. So I've got these distribution maps just to give you a little bit more information about where the native ranges of Trappa are and the introduced ranges. So we have this map on the left here with the US. In the red is where Trappa has been introduced, and then Africa and Eurasia where it's native. There's actually one report of an introduction of Trappa into Australia, and I've been checking into that. Apparently, it's literally one report. So I've been investigating that a little bit to check into that particular record of whether or not it's actually been introduced into Australia. The middle map here, if you ever want to look at the native or introduced ranges of plants in the United States, USDA has a plants database. And so you can go and you can actually look at the range maps for any number of different species. And so here's the Northeast, where you find Trappa, where it's been introduced, and up into Canada as well. And then the map on the right, this is both species of Trappa natans and Trappa bispinosa that have been introduced into the Northeast. And right down here where you have this red circle, those specific records are for Trappa bispinosa, which has been recently introduced to the US, and those are within Chesapeake Bay here in Virginia. And all of these records are of Trappa natans, which has been here since the late 1800s. And what we're finding is that this newly introduced Trappa bispinosa is spreading within the freshwater reaches of the Potomac River watershed. And we're expecting it very soon to overlap with the closest Trappa natans populations, which are actually in Bird River, Maryland. Okay, so there's two species now that have been introduced to New England. We have Trappa natans, introduced in the late 1800s. They have specific records of individual events where someone got seeds, brought them over, threw them in the closest pond, the plants grew, and then they escaped. The Trappa bispinosa inumi that we're concerned about, and it's spreading in within the Chesapeake Bay watershed, specifically it's only in Virginia at the moment, was first observed in 1985. There was a dried museum specimen that a USGS researcher found, but it's been misidentified and since that time until 2016, where it was found actually growing within the Potomac River in Virginia. And it had some morphological differences that I'm going to go into that really kind of stood out. It looks like water chestnut, but it appeared to be a different species of water chestnut. So Trappa it's actually a really beautiful plant. It's herbaceous, meaning to put it in a relative example would be if you're thinking of a woody plant, that would be like a tree. Herbaceous is going to be more like your kind of fleshy plant. Think of a tomato plant versus, say, an oak tree. It's an annual, meaning that it completes its life cycle in one growing year. It makes these really neat rosettes that float on the surface of the water. And you usually find it in, just one moment. It's found in freshwater habitats, rivers with minimal flow, wetlands, generally shallow water, 6 or 10 feet or less, even though it has been found in a little bit deeper water. On the Potomac River, it was found in, the Trappa bispinosa was found in tidal influenced water, which was interesting. The stems were just really long to be able to accept the changes in the water level elevations throughout the day. The floating rosette has these glossy leaves. I'm kind of highlighted this over here on the right, in the top right picture. They're attached to a stem. Below here, these are some seedlings of, I think those are Trappa bispinosa. You've got the floating rosette, and this is the stem here. Here's the seed, and then here are the roots. And the rosettes float because the petiole of each floating leaf actually has this spongy structure that allows it to float. So the so there's air trapped within the structures. The floating leaves are triangular. They have varying serration or teeth. It just kind of depends on the species or varieties of Trappa. Each stem can produce several branches. It actually makes a complete flower, meaning that you find both the male and the female flower parts within one flower. The fruits and the seeds are hard. They have really sharp spines. This is a Trappa bispinosa seed here. And you can see these spines on the edges on on each side of this particular, this is a germinated seed. That's the cotyledon up here. So they generally have 2 or 4 spines. Each seed can produce 10 or 15 rosettes. Those are the big rosettes here that I was talking about. And that each rosette can produce 15 to 20 seeds. And the seeds reportedly last for up to 12 years in the seed bank. And so once the seeds fall off the plant, they get into the sediment, they can hang out for a while. Although the majority of the seeds generally germinate within a couple of years of being produced. So here's a more close-up view of the abaxial, which is the top of the rosette. Here, this is Trappa natans. And you can see my hand to kind of give you a visual reference of the diameter of that rosette. And this is the bottom, the adaxial portion of the rosette. And you can actually see some fruits being produced here. There's my thumb for size reference. Trappa bispinosa. Here's the top of the rosette. There's my hand again, give you a reference of the size. And then here's the bottom. And you can see one of the wicked looking fruits of Trappa bispinosa. And then here's the stem. And the stem would be up here on Trappa natans. My finger is covering up the stem, but whenever y'all are participating in the harvesting portion of the experiment that you're going to be doing as part of a project for your class, there's some really important plant parts that you need to pay attention to. And so these are the flowers, the immature fruits, and the mature fruits. And so for Trappa natans, real pretty white flowers, white petals, 4 petals. You can have several flowers per rosette. The immature fruit, this is in the middle upper panel here. You can kind of see the formation. So these are actually the sepals that turn into, that develop into the spines. And then here's a fruit on the right panel. It has an exocarp, fleshy outer covering, and then the hard seed is inside. And then for bispinosa, we have pink petals as opposed to white petals, still 4 petals. The immature fruit here in this middle lower panel, a little bit smaller, probably just, I mean, half a millimeter or half a centimeter. And then as it grows to the right here, you can see this mature fruit. It's about 2 centimeters in length from the top to the bottom, where it's attached to this structure here, which is called a peduncle. And up here on Trappa natans, it's the same thing. It's called a peduncle. And the fruit actually attaches or detaches when it's mature at this point. I already mentioned the pink petals versus the white petals. Another interesting aspect of the morphology of the actual flower is that Trappa bispinosa has 4 to 6 anthers. So the anthers are the male portion of the flower. That's where you're going to find your pollen. And then Trapanotans generally has 4. You can kind of see the anthers here. And then you've got the stigma, which is the female portion of the flower. The underside of the leaves for Trapa bispinosa, when compared to Trapanotans, are red versus so bispinosa red, Trapanotans is green, which is a nice or a nice morphological distinguishing feature between the 2 of them. And then we have I've already showed you the fruits, but the 2 horns for Trapa bispinosa versus the 4 horns for Trapanotans. And I just want to emphasize on the left here, these are actually fruits. They still have that fleshy outer covering on. And then these are the seeds on the right. That outer fleshy covering, the exocarp has rotted off. And so you have this really hard, large, several centimeter long seed. Another interesting aspect of both of these species is the seed germination requirements. And we have 2 pictures here. The one on the right is Trapa bispinosa, the one on the left is Trapanotans. And these are what you call cotyledons. And these are the early stages of development that first emerge from the seeds themselves. And for Trapanotans, because it has been here since the late 1800s, we know that it requires a cold stratification of 4 degrees C, meaning that it needs to basically be refrigerated or go through a dormancy period in order for the seeds to germinate. There's a chemical process that it needs to undertake before that can happen. And it also requires, so in addition to the temperature, it also requires a length of time. And so that time, it can be 63 days to 107 days, or which has been found in the literature. So that is the minimum number of days that these seeds have to be exposed to 4 degrees C for the best germination. And the Trapa bispinosa, we don't really have any information on the particular species that we have introduced into the Northeast. That's a part of my dissertation work, is to find out what the ecophysiological temperature tolerances are for this species, and that will that gives us information on what watersheds and latitudes that the species could actually continue to establish in. I do know, just from some of the previous studies that I've done thus far, Trapa bispinosa does not need to be refrigerated. They will germinate fairly soon after the exocarp, the outer fleshy part of the seed falls off, and it can germinate at temperatures colder than Trapanotans. I also know that the seedlings can survive and are at higher temperatures than Trapanotans, which has some sorry, I've got something stuck in my throat. Implications for how this plant can expand its range within the US and start jumping watersheds, basically. So I'm getting close to the end of my presentation here, but so a part of my dissertation was a greenhouse experiment, and that was looking at whether or not these 2 species hybridize. And so what we wanted to do was we wanted to introduce pollen or the gametophytes from either a pollen parent to a seed parent. So going from Trapanotans to Trapa bispinosa, so pollen would be transferred from Trapanotans to Trapa bispinosa, and the seed would be produced by a Trapa bispinosa plant, or the opposite, pollen from a Trapa bispinosa plant introduced to a Trapanotans female stigma, and whether or not fruits would be produced by either of these methods of introduction of DNA. So, and we also want to know whether or not, so, I mean, who cares if they hybridize? Well, we care because we know that other species, when they hybridize, they have a tendency to become more robust, and they have a tendency to take on the traits of their parents that make them more invasive. And so we need to find out if that's going to be an issue or not. And so is the offspring going to have reduced vigor? Or not really going to do anything? Are they even going to germinate? Is it going to be about the same, or are they going to be increased vigor and something to really be worried about? And so the harvest that you guys will be participating in will be basically doing what's called a destructive harvest. We're going to be breaking down the experiment, taking the plants, processing them, taking some measurements, things like rosette diameter, the length of the stem, and a few other parameters to give us some ideas, just like on the size of the plants and how that relates back to growth. And then we'll be collecting seeds that have been produced. And all the seeds that will be produced, we're going to keep those. And I'm actually going to grow them out here in Texas, which is where I'm based. And then I'll do another experiment where I look at the actual, grow them out. How big are they going to get? How are they going to perform when compared to their parents? So very exciting stuff. But so anyway, I hope that you watched the other videos of Ben when he gave you a breakdown of the protocol for the harvest. And then, so yeah, once we get those seeds and we grow them out, I actually will be collecting DNA samples, and then we'll be actually looking to see whether or not hybridization actually occurred, in addition to any visual clues that we might get from morphology, say seeds that look like Traponatans, but they have the same number of spines as Trapobispinosa, and things of that nature. So thank you all for listening in. Appreciate it. Hope I didn't have any problems with sound or video. And thank you. And if you're curious, this is my dog Gracie, and she goes with me in the field sometimes. All right. Thank you very much. I'm going to go ahead and end my presentation.